Thank you. So I'm going to present you my thesis project, which is entitled Super-generalist Species in Complex Ecological Networks, Forms of Interactions and Implications. I have a La Caixa fellowship, and okay, so as the title says, my thesis is based on super-generalist species. These are species that within an ecological network interact with multiple group of species, since they have a big degree of interactions, and also they connect different modules of the network. So they have a central role. These species are tightly linked with the concepts, the old concepts of keystone species, that are species that have an important effect in trophic cascades, and it's also linked with the concept of foundation species. These are species that are dominant within in a community, and they have a central role since they sustain a big part of the biodiversity. And having this as a model, we have chosen the lentisc as a biological model that I imagine most of you know. This plant is present in the Mediterranean shrubland, and it dominates a big part of the community, and it sustains a lot of the species that are present in this ecosystem. And the lentisc holds interactions along all its life cycle. During the vegetative growth, it interacts with a gold-forming aphid, or Planura lentici, which is specific of the lentisc, and also with many insect herbivores that feed on the leaves of the lentisc. Then the pollination is anemogamous, so it doesn't have pollinators. But during the pre-dispersal phase, when it's forming the fruits, there is a wasp called Megastigmus Pistaciae that oviposits in the fruits, and the larvae of the wasp feeds on the embryo, causing unviability of the seeds. Then during dispersal, it interacts with many avian species. Some are antagonistic, as finches, green finch or finch, and also mutualistic species that are legitimate dispersers, which are a lot of species, and they provide an important food source. And lastly, when the seeds are dispersed, they encounter predation by rodents that also act antagonistically. So it holds different types of interaction that range from mutualism to antagonisms. Having as a model the lentisc, the idea of the thesis is to understand how super-generalist species organisms shape the interaction network, their evolutionary implications, study the generalist strategy, and their implications in the ecosystem functioning. And we will do this by targeting through 4 chapters. The first chapter looks at the effectiveness, which is a concept that I will introduce later on. But the idea is to look at the effectiveness that the two partners provide for each other during the interaction. Then the chapter 2 would be based on individual-based networks that will be based in specific individuals of lentisc, and using traits, individual traits of these plants. The idea is to see how they shape their interactions in a network. The third chapter will be more holistic, because the second chapter will be based on dispersal, so only the mutualism part. But the third chapter would include also antagonistic interactions. So the idea is to having all these type of interactions put them together through a multi-layer approach that I will explain also later on. And the fourth chapter would be to explore the idea of the super-generalist strategy. And this will be done by using other examples of individual-based networks from other studies in comparison with the lentisc. So, at the moment, we have 2 populations of study, both in Doñana, one in Laguna de las Madroñas in the reserve, and the other one in La Mancha del Rabicano in El Puntal area. In each population, I have 4 lentisc marked, 4 in each, and in the Mancha del Rabicano, I also have other experiments going on that I will explain. So for each individual of lentiscs that we have marked, we have individual data, extrinsic data of the location and the neighborhood surrounding it. We have also biological data on the area, the perimeter, the height, and the structure of the plant, whether if it's more like a tree or more like a shrub. We have measured the crop size at the beginning of the season, so how many fruits it has. The viability, since the lentisc also is very interesting because it has, the fruits of the lentisc has two colors, the black one, which is when it's ripe, and the red one that is more in the development. Even they can even, sometimes ripe fruit can also be red. But also the lentisc, the viability of the fruits is very low. It's around 20%, but it can vary a lot. And the causes of unviability are the wasp that I explained before, parthenocarpy, and abortion. So there's three main causes of fruit unviability. And we have this measured for each individual along the fruiting season. Then also we have seed traps like this one that are settled under the plants, and there we are collecting all the seeds all the fruits that fall from the plant, as well as predated seeds and dispersed seeds from the birds that are visiting the plant. With this information, we can calculate a proxy of the effectiveness, a proxy of the fitness in dispersal. That will be based on the initial crop size, so how many fruits the plant has in the beginning, all the fruits that have fallen along the season, which will be calculated using this data, and also we subtract this to the predated seeds, and we have the number of seeds that are dispersed that can be a proxy of fitness, of dispersal fitness. Then the second most important data that we are collecting is the interactions. So the frugivore interactions are measured in three ways. First one, and maybe the most important one, is through DNA barcoding. So all the seeds dispersed, and as well as any feces that falls in these seed traps, are analyzed later in the lab to identify the bird who they belong to. We also have monitoring cameras that are settled for each plant along the seasons 2 hours, so in several times. And we also have direct observations in the field. This will be all the frugivore interactions. Then the herbivory damage that we measure in the spring when the new sprouts appear. And the idea is, I've already done some, and the idea is to categorize the herbivory damage in several categories and associate these damages to a functional group of herbivores. The seed wasp predation is already measured in the seed viability experiments that I explained before, which are how the fruit looks when it is when the wasp deposits its larvae within. And the last interaction data will come from rodent censuses that we are carrying on at the moment, and also maybe next year, and where we see the appearance of these animals, like moose or apodemus and rat, in the individuals on the lentiscs. Then, since we aren't also interested in the seed fate or where the seeds appear, we have categorized 5 types of microhabitats where the seeds can fall. One is under lentisc. The other one is in FR is fruiting species, so under plants that have fleshy fruits. NF is non-fleshy fruit species. PP is from Pinus pinea, so under pine trees, and in the open area. And for each of these categories, we have 15 replicates, except for open area, where we do open transects because it's very hard to obtain seeds in this microhabitat. And then we have for each microhabitat we have seed traps also, where we are collecting all the seeds, and we will do barcoding as well as we have observations for how the birds use these different kinds of microhabitats. Then also for each microhabitat we have seed predation experiments where we where we see what is the predation risk for the seeds in the different types of microhabitats, having a control to exclude the rodents. And then we also have germination experiments to see the germination of the seeds in the 5 different microhabitats. And lastly, also we have data of the area on vegetation transects to know all the species that are present in the plot. We also carry on weekly or fortnightly avian censuses to know the bird abundance. And we also have data on temperature, humidity, etc. So, with all this data, I just want to show you a preliminary result of what I've recorded so far. These are the barcoding samples that we have collected, which is around 5,500. These are from this year that we are doing. We're still going on with the field season, and these are from last year. Last year I started a bit late, so that's why this is shorter. But we have already analyzed all the plants, all the seeds that have fallen under lentisc. And all this data will be useful for all the chapters that I am going to target. So, and this is for you to have an idea of all the avian species that have appeared in the barcoding. As you can see, the robin and the Sylvia melanocephala are the main consumers of lentisc, at least during the winter season, because this is only for winter data. So for the first chapter, which is based on effectiveness, is based on a concept that I will try to explain. The idea of measuring effectiveness is a way to calculate the effectiveness that the one partner provides for the other. So normally these have been measured from a photosynthetic approach, where they study the effectiveness that different dispersants provide to the plants. And this is, well, the effectiveness concept is based on 2 components: the quantity component, so it's the number of seed dispersed in this case, and the quality component, which is the quality that the disperser provides for the plant. And this can be, this is something that we've done during my master's thesis, and is to use this framework also from a photocentric approach, that is, measuring the effectiveness that different fruits have for the disperser, for the consumer. So in this case, the quantity would be the same, also the feeding rate of the animal, so the quantity of fruits consumed, but the quality in this case would be the energy that the fruit provides for the bird. And for the first one, the phytocentric approach, we have data on how the different species, how much they consume for each plant. And for the quality, to calculate the quality that each bird provides to the plant, we know how much they use the microhabitat use. We know the predation and the germination probability of the seeds that finish there. And with this accumulation probability, we can calculate the quality that the different seed dispersers provide to the plant. For the zoocentric approach, then, we have it would be to calculate the effectiveness that different plants, among them the lentisc with other species present in the Mediterranean shrubland, how effective are for some of the species for some of the main species. So we have data again on fruit consumption that comes from the seed traps, as well as from data that also Pedro has from his thesis. And then we know the fruit energy from studies that have been carried here since 1987. And then, well, this is how an effectiveness landscape looks like. The quality component, the quantity component is here. So here are how many seeds are consumed, and here is the quality of the dispersers. In this case, each point represents an animal or a bird, while in this case, each point would represent a plant. So the idea is to look at the dependencies. So for example, if this was a lentisc, if for example a robin is very effective since it has a big quantity component and a big quality component, if in turn the lentisc in comparison with other fruits has also a high effectiveness for the robin. So we want to see if these dependencies are symmetric or asymmetric. The second chapter will be based in individual-based networks that we We have data on this data is represented in an adjacency matrix, where we have here the plants and all the records of species that are consuming it. And we know for each plant also individual traits. So this is a preliminary network that I've built with the data we have so far. Each blue point would be a plant, and the rest are the bird species. And since we have information on individual traits, as I said, the idea is to use an exponential random graph model to see how these traits may be accounting for the structuring of the net, to see if they're affecting the way the individuals shape their interactions. This is non-weighted, and this would be a weighted matrix. I just wanted to show you that, well, these are the plants, and these are the bird species. These are weighted, so as you can see, the robin, for example, is one of the main consumers, but it interacts with most of the plants as well as the sylvia, while, for example, other species as the Genopica, it interacts only with a few of the individuals, so it doesn't interact with all of them. Okay. Also, an idea is that since the lentisc has a very long fruiting season that goes from August to February, more or less, the pool of species that are feeding on the plants changes if there's a species turnover along the season. So the idea is to compare, so to see how the network changes in the beginning of the season, when the sub-Saharan migrants are present and are feeding on the plant, on the fruits, and when they leave, the European migrants arrive as the robin, and later on, turdid species as Tyrtus philomelos arrive, and this may change the structuring of the net. So the idea is to see how this develops along the fruiting season, and if there is changes in the structuring of individuals, how it holds its interactions. So the third chapter would be more looking at a more holistic way. So in this case, I would try, the idea is to include the mutualistic network that I presented before, together with antagonistic interactions. This would be pre-dispersal predation by the wasp and the finches, the free dispersal, the post-dispersal predation by the rodents, and the herbivory interactions. Each network will be connected through the individuals, since the individuals of the plants are present in all the layers, and they can be connected by this internode link. This will be a multi-layer approach, and these plants are connected in all the layers. Yes, okay. And then last, the fourth chapter is comparing the individual base network of the lentisc. So the idea of this chapter is based on niche variation. What we want to see here is how a generalist species, the individuals within it, how they behave. If they behave like a generalist species, like a generalist individual that is using all the resources present, or otherwise the individuals behave as specialists. So even having a big niche, they only exploit a part of this niche. And this will be done by using other individual-based networks of other species that we have that are published or not. For example, well, the Pistacia one, but also Cecropia or Prunus mahale, which also there's data about. Juniperus, that my colleague is working with, also in an individual approach. Chamaerups, etc. So the idea is to take all these nets and see how the individuals are behaving in the networks, and explore this idea. And well, last, I just want to show you the tentative schedule. My fellowship started in December 2018 and will finish by December 2021. I'm already in the second part of the field season. I'm finishing already the second year. We will have a laboratory analysis of the DNA barcoding that we have already done some, but to finish all the samples. We also have planned an academic stay in the beginning of 2021, and the idea is to deposit the thesis in June of 2022, more or less. So thank you very much for your attention, and thank you to all the people in the lab that has helped me with the thesis and will help me also.